Welcome back to this next video in which we are discussing influenza. So, we are in the process of discussing the influenza viruses, and we've seen that there are three different species of influenza virus: the influenza A virus, the influenza B virus, and the influenza C virus. And we're currently in the process of discussing the structure of the influenza A virus, and later we'll talk about the influenza B virus and the influenza C virus. They're not radically different, but there are differences nonetheless. So, let's just summarize where we've got to so far. We have seen that the influenza A virus is a enveloped virus, and that in the viral envelope there are three different types of protein, namely the hemagglutinin protein, which is involved in binding to the initial host cell, the neuraminidase protein, which is involved in viruses getting out of the host cell, and then the M2 ion channel, which is also involved in the entry mechanism of the virus. Underneath the envelope, you then have this M1 shell that is known as the matrix of the influenza A virus. And this is made up of around 5,000 M1 proteins, and each one of these blobs is an M1 protein. Then inside of that, we have the ribonuclear proteins, of which there are 8 inside each influenza A virus particle. And by the way, a figure that I don't think I ever gave you in the previous video is how big these little things are. So what is the diameter of an influenza A virus? It's usually between 80 and 120 nanometers. So they're very small little things, but that's roughly the size of these things. That's the size scale that we're dealing with here. So much, much smaller than a human cell. Okay, so inside the M1 matrix then, we have the ribonuclear proteins, of which there are 8, and these ribonuclear proteins, each of them contains a single-stranded piece of RNA that is part of the viral genome. The viral genome is a segmented genome into 8 different pieces, and all of the genetic information is stored in single-stranded negative sense RNA molecules. Now, we've seen that each of these ribonuclear proteins, as well as containing the single-stranded negative sense RNA molecule, also contains a huge number of the nuclear protein, which makes up the capsid of each of the viral genome RNA molecules. And then associated with it, you've also got one of these RNA-dependent RNA polymerase enzymes, which is made up of 3 separate proteins: the PA protein, the PB1 protein, and the PB2 protein. Finally, also inside the matrix of the influenza A virus particle, you have some of the non-structural protein 2s, or the nuclear export proteins, which are these things here. Alright, so where were we? Where were we going to continue on to? We were going to now discuss the structure of hemagglutinin in a little bit more detail. Not in massive detail, but in a little bit more detail than we have done previously. We're also going to discuss Neuraminidase structure in a bit more detail, and also the M2 ion channel. And then I'm going to talk to you about the H1N1, H5N1, H2N2, H3N2, all of that stuff. That classification of influenza A viruses. So, to business then, let's discuss the hemagglutinin protein then in a little bit more detail. So, each of these spikes then that we have in the viral envelope here, it's not just a single polypeptide, it's actually made up of multiple different polypeptides. So we have seen that viral genome segment 4 contains the gene for the hemagglutinin polypeptide. Each one of these hemagglutinin proteins, however, in the envelope does not just contain one of those, it's actually a trimer of those. So let's do this. So when you actually first synthesize a hemagglutinin protein, it is in the form that we would call HA0. So when you first actually synthesize the protein that is coded for by viral genome segment 4, it produces us a polypeptide which we call hemagglutinin 0. And this is going to make the hemagglutinin proteins that are on the viral envelope surface. However, in order to actually make one of these molecules in the viral envelope, we need to take 3 of these, we need to trimerize them together, and we also need to cut each one of them that is in this trimer into 2 separate portions. So hemagglutinin 0 is cut into 2 separate portions: hemagglutinin section 1 and hemagglutinin protein 2. So really, the hemagglutinin end molecule that is actually in the envelope of the influenza A virus particle actually consists of 6 separate little pieces, 3 hemagglutinin 1 polypeptides and 3 hemagglutinin 2 polypeptides, although they were both synthesized together as this hemagglutinin 0 polypeptide. So let me now actually attempt to draw a more in detail picture of this. So here, this is going to represent the phospholipid bilayer of the viral envelope, and you can tell that I've zoomed in because I'm now actually drawing 2 lines for the phospholipid bilayer representing the 2 sides of the lipid bilayer rather than just 1 line as it was previously. Now, let me put the hemagglutinin in, and we'll stick to drawing it in red here then. So, the hemagglutinin 2 polypeptides, these are the ones that are actually going to span the viral envelope, and the hemagglutinin 1 polypeptides, they're going to be more globular and they're going to be sat on top of the hemagglutinin 2 polypeptides. They still remain bound to one another, that's important. When we cut the hemagglutinin 0 into hemagglutinin 1 and hemagglutinin 2, even though they're no longer linked by the peptide link anymore, and just to make this absolutely obvious, let me just draw a little bit in. Here, this can be the hemagglutinin 0. We then cut it up, and we're left with hemagglutinin 1 and hemagglutinin 2. So even though they're no longer a single polypeptide, they're no longer linked by this peptide link, they're going to bind using their R groups of amino acids in their length. And indeed, they're going to bind together by disulfide bonds, which are a very nice, powerful bond. It's a nice covalent bond that you can form between 2 polypeptides. And so let me now draw the picture here of the hemagglutinin spike then. So here are the 3 hemagglutinin 2 polypeptides to start off with. And I'll draw it in a nice sort of cylinder shape here. Okay, so let me split it into 3 separate portions then. So here we go. I'll put another line down there. Okay, so what I'm trying to display here is 3 hemagglutinin 2. So this is 1 hemagglutinin 2. Here is another hemagglutinin 2. All the way down here, and then there's a 3rd hemagglutinin 2 over there. So all of these are representing the hemagglutinin 2s. And now, bound on top of the hemagglutinin 2s, and we're imagining this is the inside of the virus facing towards the M1 proteins, and this is the outside of the virus. Sat on top of the hemagglutinin 2s, on the outer side, are going to be the hemagglutinin 1s. So I'll do these in a separate colour. So we'll do it in green to have the most radically different colour from red. So, bound on top then, here is a hemagglutinin 1 on top of that hemagglutinin 2, here's another hemagglutinin 1 on top of its hemagglutinin 2, and here's the 3rd hemagglutinin 1 on top of its hemagglutinin 2. So I hope this is now clear. We start by synthesizing 3 hemagglutinin 0 polypeptides, we trimerize them together, we then process the hemagglutinin 0 polypeptides in the trimer so that they're actually split into these 2 components, each of them, hemagglutinin 1 and hemagglutinin 2. However, they still remain bound together, just not by a peptide link anymore. So we've got 3 dimers of hemagglutinin 2 with hemagglutinin 1. So I'll just label up the hemagglutinin 1s here. So all of these green blobs, they are representing hemagglutinin 1 polypeptides. And the whole thing then is the hemagglutinin molecule that I drew here. So this red spike, this is what it actually is. It's trimer of these dimers of hemagglutinin 1 molecules bound to hemagglutinin 2 molecules. Now, I said that hemagglutinins' role in the viral replication cycle was going to be in the initial entry mechanism. It binds to molecules on the surface of host cells, and we'll talk about what molecule they bind to much later on when we come on to the viral replication cycle. But for now, let me say that it's going to be the hemagglutinin 1 polypeptides that actually have the binding site. So I'll just put this here in yellow. So these little yellow spots here, these can represent the binding sites. So I'll just put that down here. So these are representing the binding sites for whatever the ligand is that's on the surface of host cells. So the important message that I want to get across here is that each of those hemagglutinin spikes on the surface of the virus particle has 3 binding sites for the molecule that is on the surface of host cells that the virus can bind to. That's the important message that I hope I've communicated with this picture. Okay, So, there is hemagglutinin. Next, let's do neuraminidase then in a little bit more detail. So, just as hemagglutinin, just as this hemagglutinin spike was not just a single polypeptide, it was actually made up of multiple hemagglutinin polypeptides coded for by genome segment number 4. Again, this neuraminidase structure, this mushroom structure that I've drawn here, is not a single neuraminidase polypeptide. Instead, it's a tetramer of neuraminidase polypeptides. It's slightly more simple though, because we're not going to cleave the neuraminidase polypeptide into 2 bits. So, viral genome number 6 codes for the neuraminidase polypeptide. You need to make 4 of them and then bind them together, tetramerize them to make a neuraminidase protein complex, which is going to be what these things on the surface of the virus particle are. So, let's again draw this to make it utterly crystal clear. So, just move this up a little bit. Again, I'll draw the phospholipid bi there, here. And now let's draw the neuraminidase, and I'm going to try and get it in a mushroom shape, as mushroom shape as I can possibly get it. So, here we go. This is the top bit of the neuraminidase tetramer here, like so. And then here is the portion that can then go down through the membrane and implanting it into the viral envelope. I'll just try and make it look a little bit more 3D, like so. Okay, so this is my zoomed in neuraminidase, and now what I need to do is separate it into 4 separate portions. So this isn't too difficult. So here we go. I've now separated it into 4 separate portions. Let's just continue on the separations downwards, like so. And we'll put another separation in there. So I hope this picture is clear. We now have 4 separate neuraminidase polypeptides shown here. Here is one of them here, like so. Then there's another one back here, which includes this bit here. In fact, I might just try and colour in one. So I'll get a thicker pen here. So in red, to make it utterly clear, this one that I'm colouring in, this is a single neuraminidase polypeptide, and of course it also includes that aspect there. So that's a single neuraminidase polypeptide. You've got 4 of them in total making up the neuraminidase protein complex. Now, neuraminidase is an enzyme, also called sialidase, and again, we will discuss in more detail later when we come onto the replication cycle precisely what reaction it catalyzes. But it does catalyze a reaction, and that reaction is very important for virus particles being able to bud off the infected cell. Now, what important lesson am I hoping to give you here? Why have I bothered telling you that these neuraminidase protein complexes are tetramers? The important thing that I want to communicate is that each one of the neuraminidase polypeptides in the neuraminidase tetramer has its own active site. That's the important message. So I'll get another colour again. We'll have blue, I think. And now I'll draw on these active sites. So this blue circle is representing the active site of that neuraminidase polypeptide. This blue circle is representing that one's, and then that one's. Okay. So what I have drawn on here is each of the 4 neuraminidase polypeptides in the neuraminidase protein complex has its own active site where it can catalyze this reaction. So each neuraminidase protein complex is actually, it's got 4 active sites for catalyzing this reaction. So it's not just capable of catalyzing one reaction at once, it's capable of catalyzing 4 reactions at once. That's the important message I wanted to get across here. So that's the structure of neuraminidase. Just for completion, I'm now going to go over the structure of the M2 ion channel in a little bit more detail. Okay, so again, the M2 ion channel is a tetramer, as ion channels often are. They're not usually monomers, ion channels, they're usually multimers, and tetramers is a common number of protein subunits to have. So again, here we go, this is going to be the phospholipid bi there, zoomed in here, and now what I want to do is draw on the 4 subunits of the M2 ion channel. So just to remind you, M2 is coded for by the 7th viral genome segment along with the M1 protein, so we need to make 4 of those M2 polypeptides stick them together, and then we'll get an M2 ion channel, which is what I'm just about to draw here. So I'll have it in a nice cylinder shape here, like so. Okay, so here is the pore down the middle, and I'll try and make it look a little bit better. And here are the 4 separate subunits that make up the entire M2 ion channel here. Okay, so each one of these is an M2 polypeptide, or an M2 protein, and together the entire thing is the M2 ion channel. Now, that's a bit of a boring picture. It's a bad picture as well. Let me give you a little bit more information about this channel. So we said earlier on that this is an ion channel for protons, so it's going to conduct hydrogen cations. Now, what activates it to open? Is it permanently open, or does it get activated by something? And the answer is it is not permanently open. It gets activated by something. And the thing that activates it to open is protons, a high concentration of protons. Now, which side of the membrane do you have to have the high concentration of protons on then to get the protein to change conformation and adopt the open conformation? Well, it's the outer side. So, just to orient you, this is the outer side of the membrane facing the outside of the virus particle, whereas this is the inside facing into the center of the virus particle. So you'll have the M1 proteins here, and the inside of the ion channel, remember, empties or opens in to the inner portion of the virus, so it will conduct protons into the center of the virus particle. I'll just get the picture back here. So remember, if protons were to come in through here, they would come right into the core. They wouldn't get stuck in this space in between the membrane and the M1 proteins. They come right into the core here. They bypass the matrix. Okay, so it is a high proton concentration on the outer side of the membrane that activates the ion channel. Now, of course, a high proton concentration corresponds to a low pH. Remember, they're related inversely. So when the pH goes down on the outside of the viral membrane, that is what activates this ion channel to then open. And of course, if you've got a low pH on the outside, that means you're going to have lots of protons on the outside. So which way do you think the protons are then going to move? Well, of course, they're going to move from the outside to the inside. So when you've got a high pH, made the classic mistake, a low pH, a high proton concentration on the outside of the viral envelope, it activates the M2 ion channels to open, and protons will then move from the outside to the inside, and of course they're going to cause changes on the inside of the virus, but we'll come back to that later. There's just a little taster of what will happen later on. Okay, so there's my more in-depth discussion of the M2 ion channel. So enough of proteins. Let's now talk then about this HN stuff, the different serotypes of influenza A viruses. Okay, so this is complicated to understand, and the main reason that it's complicated to understand is that it's not really real. It's something that someone very clever in a lab created as a classification structure. Okay, and really to understand the classification structure properly, you need to know all the experiments and understand all of the experiments that they were doing and which they used to actually come up with this classification, and I do not understand all of those experiments, but I'm still going to try and explain this classification system. Okay, so influenza A virus, we'll forget about IBV and ICV at the moment, we're just talking about influenza A virus. We'll come on to influenza B and influenza C. Influenza A virus, it's a species of virus, and just as I said at the start, just like the human species, it has loads of different members, loads of different strains that are all quite different from one another, just like there are loads of different humans that are all very different from one another. Now, we have a way of classifying the strains of influenza A virus. There are absolutely loads of strains of influenza virus, a huge number, but we can group them together into these classes, which are known as the serotypes of the strains of influenza A virus. So every single different strain of influenza A virus has a serotype. So let me just bring this up here. So we're now going to discuss serotypes. So every single strain of influenza A virus, if you get me any influenza A virus in the world, bring it to me. In principle, I can work out this thing called the serotype. And this concept is a way of classifying influenza A viruses into groups, and the way that it works is by looking at the structure of the hemagglutinin proteins and the neuraminidase proteins of the virus. So remember, these viruses are all different. All the different strains of influenza A viruses are different. They'll have slightly different genomes, slightly different combinations of organic bases in their RNA molecules, and that means that they will make slightly different proteins. So their hemagglutinin proteins and their neuraminidase proteins, and also all the other proteins here, will be slightly different between the different strains. We don't care about all the other proteins as far as this classification is concerned. We just care about hemagglutinin and neuraminidase. And why do we care about those so much? Because the way that this classification system works, the way that the experiments that are used to actually work out what class an influenza A virus should be put into work, is by looking at the antibodies that can be produced by a host in response to the influenza A virus. So remember, one of the key immunological mechanisms to defeat the influenza A virus is going to be that we produce antibodies. We get a B cell response and we produce antibodies against it. We'll talk about all of this in much more detail later on. We don't need to understand the B cell response much in order to discuss this. We just need to know that antibody molecules, these special molecules that are capable of binding to proteins of the pathogen and which are directed specifically against the proteins of that pathogen, can be produced in response to the pathogen infection. Okay, now, two of the key proteins that are going to get antibodies produced against them in the influenza A virus are, of course, the hemagglutinin protein and the neuraminidase protein. Why? Because they are so important, and also because they are nicely on the surface of the virus particle. And when we have proteins nicely on the surface of the virus particle, they are perfect targets to produce antibodies against. Now, when you have two different strains of influenza A virus that have kind of similar hemagglutinin molecules and neuraminidase molecules, the antibody responses that you would get to those two different strains will kind of overlap. So remember, when you get, for instance, an antibody response to hemagglutinin, you don't just produce one type of antibody, you produce loads of different antibodies. And I think at this point, it will probably be helpful for me to have a picture of an antibody. So let me just draw one here. So remember, a very basic picture of an antibody is what I'm drawing here. And the molecule, remember, consists of 4 polypeptides joined together, and really you have a dimer of dimers. So both of these molecules here in the Y, these are the immunoglobulin heavy chains, or the antibody heavy chains. Remember, immunoglobulin is the fancy word for antibody, so I'll put some of this terminology down here. So immunoglobulin is the more fancy term for an antibody molecule. So this is equal to antibody. And the molecule, a very simple picture of it, I've drawn here, and it consists of these 2 chains. We've got the immunoglobulin heavy chain, and Ig is short for immunoglobulin heavy chain, and we've got 2 identical immunoglobulin heavy chains there. This one here, and this one here, and I think I'll try and change the colours here, so I'll go over in a separate colour. So in purple here, this is the and I'll get a thicker pen so it actually does cover the previous colour. So here is one immunoglobulin heavy chain, and then here is another immunoglobulin heavy chain. Oh, what a mess. Okay, so those are the immunoglobulin heavy chains, and then we've also got 2 identical immunoglobulin light chains, which are the ones that are left, whoops, I haven't changed colour, in this other colour. So those are the immunoglobulin light chains. So to make an immunoglobulin then, you need 2 copies of an immunoglobulin heavy chain that are identical and 2 copies of an immunoglobulin light chain, and you join them together in this way. And then right at the tip here, this is where the molecule is capable of binding to some antigen, which is just a name for any protein that is capable of having an antibody produced against it. So you get 2 identical antigen binding sites in any immunoglobulin molecule. Now, for different antigens that you might want to produce antibodies against, you're going to have different antigen binding sites, and therefore you have to have different immunoglobulin heavy chains and different immunoglobulin light chains. And that's the miracle of the adaptive immune response, that it is capable of producing a huge number of different types of immunoglobulin heavy chain and a huge number of different types of immunoglobulin light chains. Therefore, it's capable of producing loads of different antibodies with different antibody binding sites, so it's capable of launching a response against loads of different antigens. Okay, so when we launch an antibody response against hemagglutinin, there are loads of different sites on the hemagglutinin molecule that you could get antibodies binding to, and this has a special name, this concept. This is the concept that an antigen has multiple different epitopes. So the epitopes of an antigen molecule are all these different little portions of the molecule that, are different in their chemical nature. They have different functional groups exposed, etc., that you could produce antibody molecules that would be capable of binding there. So, the hemagglutinin molecule has multiple different epitopes, and therefore when you get an antibody response to it, you get loads of different types of antibodies being produced that can bind to the different epitopes. So, coming back to my classification of influenza A viruses then into different serotypes, the 2 different strains of influenza A virus that I have and want to classify, that if they have similar hemagglutinin proteins, they will have overlapping antibody responses against them. So I can draw this with a little bit of a Venn diagram here. So if this is one influenza A virus strain's hemagglutinin molecule antibody response, so let's say that each dot in this bubble represents a type of antibody that's been produced against the hemagglutinin molecule, and then let's have another circle then. So here's another circle, and then in here, every dot represents a antibody molecule, a type of antibody molecule against a certain epitope of this strain of influenza A virus's hemagglutinin molecule. The idea here is, look, some of them overlaps. You get some antibodies that will be capable of binding to both hemagglutinin molecules because they have similarities, they have epitopes that are the same. So when you have 2 similar hemagglutinin molecules, some of the antibodies that are produced in the 2 different antibody responses to those 2 different hemagglutinin molecules will overlap and will be able to bind to both hemagglutinin molecules because the epitope of the hemagglutinin molecule is the same. This is why this classification is called serotype, because serotype refers to the type of antibody response that you're getting in the serum of the blood of some infected host. So, how does this all work then? So basically, this classification of influenza A virus strains into serotypes is all based on how similar hemagglutinin molecules influenza A virus strains have and how similar neuraminidase molecules they have. So overall, there are 16 different serotypes of hemagglutinin molecules and 8 different serotypes of neuraminidase molecules. So this is the really important bit here, so I'll just write this down. So for hemagglutinin, there are 16 different serotypes. So we divide all of the hemagglutinin molecules of every single influenza A virus strain in the world into these 16 different categories, and each one of these categories contains loads of different hemagglutinin molecules which are all reasonably similar and therefore have reasonably similar antibody responses. They won't be the same and they won't have identical antibody responses, but they have similar enough antibody responses that in whatever experiments the people who came up with this classification use, the antibodies of the different against the different hemagglutinin molecules in a single one of these types can cross-react enough to get a positive result in the experiment, if you understand what I mean. Really, to understand this properly, you need to know exactly what experiments they were doing to try and test whether the antibodies against one hemagglutinin molecule combined enough to another hemagglutinin molecule from a different strain of influenza. However, I don't know the technicalities of the experiments that they were doing. But overall, what actually happens is that every single strain of influenza A virus is put into a category. So we look at its hemagglutinin molecules and we put its hemagglutinin molecules into one of these 16 categories, and that is based on how similar its hemagglutinin molecule is to the other hemagglutinin molecules in that category. So all of the hemagglutinin molecules in a single category are all similar enough to one another that they have similar antibody responses. Okay, and it's the same for Neuraminidase. Neuraminidase, however, has fewer categories. Neuraminidase has 8 classes. So there are these 16 different classes of hemagglutinin molecules and 8 different categories of neuraminidase molecules. Each of these classes contains a huge number of hemagglutinin molecules from a huge number of different strains of influenza virus that are all different, but they're similar enough to be classified together, and they have similar antibody responses that would overlap in the way that I've shown here. Now, of course, with lots of hemagglutinin molecules, we will get a tiny bit of overlap. The technicality that I do not know and do not understand is how big an overlap do you have to have in order to be put into the same class. I don't know the technicalities of how that works. But overall, what I do understand is that we separate the hemagglutinin molecules into these 16 different categories and neuraminidase molecules into these 8 different categories. So this is what the H and N of the influenza strains refers to. It means that every single influenza A virus strain can be given a serotype. And the way that we do this, or the way that we name them, therefore, is we just put H, and then we put a number, and then we put N, and then a number. So to give some examples, so in terms of some famous influenza viruses, so in 1918, so we'll do some famous examples. So in 1918, the world suffered a horrendous influenza pandemic that was caused by a strain of influenza A virus called Spanish flu. And this overall killed, I think, around 50 million people in the world. It was a horrendous pandemic of influenza A virus. This Spanish flu, it killed more people than were killed, for instance, in World War One. This specific strain of influenza A virus, this was in the category, the class, the serotype H1N1. Now, the key thing that I want to try and get across here is that this does not specify the actual strain of influenza virus. That's a common misunderstanding, that H1N1 is a single strain of influenza virus. It's not. It's the serotype. And loads of different influenza A virus strains will all have this same serotype. And if they have the same serotype, it means that their hemagglutinin molecules and their neuraminidase molecules of those strains that are in the same serotype are reasonably similar to one another, and their antibody responses would overlap to a significant enough degree to produce a positive result in whatever experiments the people who came up with this classification used. Okay, so there's an example. Spanish flu was a strain of influenza A virus, and it had the serotype H1N1. Let's do some more famous examples. So, give me the pen back. So, next one. In 1957, there was another horrendous pandemic. There have been 3 pandemics in the 20th century and 1 so far in the 21st century. So, in 1957, there was a horrendous influenza A virus pandemic again, which was Asian flu this time. And Asian flu had the serotype, it had a different serotype, it was H2N2. So I should have said, actually, when I was going over these 16 different categories of hemagglutinin molecules and 8 different serotypes of neuraminidase molecules, that these serotypes are given really boring names. They're H1, H2, H3, H4, all the way up to H16, and again for neuraminidase it's N1, N2, all the way up to N8. So Asian flu, this strain of influenza A virus, its serotype was H2N2. Its hemagglutinin molecules were in the serotype 2, and its neuraminidase molecules were in the serotype 2. Again, I'll stress the really important lesson that I want you to grasp from this, which is that that does not specify the strain. When you say H2N2, that does not instantly mean Asian flu. Asian flu instantly means H2N2. However, there are other strains of influenza A virus that also have this serotype, so it is not a single serotype. It's not a single strain. Okay, next famous example, one final one we'll do, the final pandemic of the 20th century, which occurred in 1968 and was Hong Kong flu. This had the serotype H3N2. So I hope that by doing these famous examples you have got the grasp of the H and N serotyping of influenza A viruses. So just to absolutely nail this lesson home, because it's something that people get very confused about, and it is confusing because really, as I say, to properly understand it you would have to know the technicalities of these experiments that they actually do to categorize or to classify each influenza A virus into serotypes for hemagglutinin and neuraminidase, which I don't actually know. Each strain of influenza A virus can be given a serotype of its hemagglutinin molecules and a serotype of its neuraminidase molecules. And these are classes of loads of similar hemagglutinin molecules. So each serotype contains a huge number of different hemagglutinin molecules or a huge number of different neuraminidase molecules from a huge number of different strains of influenza A virus that are all different to one another. They have different structures but have similar enough structures that their antibody responses are similar and they're classified into this single category. Okay, so if you give me a strain of influenza A virus, I can, in principle, do some experiments that are looking at its hemagglutinin molecules and its neuraminidase molecules, and I can say which serotype its hemagglutinin molecule should be in, which serotype it fits best in, which ones it's most similar to, and which ones its antibody response in a victim or some host are most similar to. And I can do it for the neuraminidase molecules as well. And that's what the H and the N that people always refer to when they're talking about strains of influenza A viruses actually means. Okay, so there we go. We have talked about the H and the N serotyping of influenza A viruses. The final thing that I want to talk about is the fact that of all the different strains of influenza A viruses that are on the planet, different strains can infect different species. So some influenza A virus strains are actually capable of infecting humans, but not all of them. So let me draw one final diagram here. So I'll do it in green here. So if this box represents all of the influenza A viruses on the planet, so this is all of the influenza A viruses. Okay, so let's draw the bunch that can infect humans over here. So this subset of all of the influenza A viruses, this is the subset that can infect humans over here. So not all of the influenza A viruses that are on the planet can actually infect humans. And most of the influenza A viruses strains that are in here, so I'll draw the little dots here. So each dot represents a strain of influenza A viruses. Each of those dots, most of them, each of those dots will have a serotype. It will have a hemagglutinin serotype and a neuraminidase serotype, and most of the strains that can infect humans are of the serotype either a hemagglutinin serotype between 1 and 3 and a neuraminidase serotype between 1 and 2. So most of these strains will have the serotype H1 to 3, N1 to 2. So those are the normal serotypes of human strains. Some of the human strains will have serotypes outside of that, but most human strains of influenza A virus have a serotype H1 to 3, N1 to 2. Now, most of the influenza A virus strains in the world actually infect birds. Birds are the major victims of influenza A virus. There are a huge number of strains that infect birds. So let me now put on, I don't know how to do this, we'll do this in purple here. This box can represent all the strains that are capable of infecting birds. There are a huge number of different strains of influenza A virus that are capable of infecting birds, and you'll see that there is an overlap there. Some of the influenza A viruses that can infect birds can also infect humans, but not that many. Now, in this great big subset of influenza A virus strains that infect birds, you'll have all sorts of different serotypes, so you'll be able to find every single one of the serotypes that you could possibly come up with there. So birds are really bad victims to flu. There are loads of different strains of influenza A viruses capable of infecting birds and of all the different serotypes. Now, what's one of the key things that I want to put on this picture? What the key message is now a final species that can be infected by influenza A viruses, and when I say it's the final species that can be infected by influenza A viruses, it's not. It's just the final one that I want to put on this picture. Loads of other species, such as non-human primates, dogs, horses, they can all be infected by influenza A viruses as well, but I'm not going to put those on this picture because they're not important. The important one that I want to put on this picture is pigs, and I'll put this like so. So this purple box here, this represents the strains of influenza A viruses that are capable of infecting pigs. And what's the key thing that I want you to take from this? Look, there's a big overlap between the strains of influenza A viruses that are capable of infecting humans and capable of infecting pigs. The other key thing to note is that there is a big overlap between the influenza A virus strains that can infect birds and the influenza A virus strains that can infect pigs. That is important, that pigs can be infected by a lot of influenza A viruses, and they could potentially be infected at the same time by a human influenza A virus strain and a bird influenza A virus strain. And that's important because when the pig gets infected by a bird strain and a human strain at the same time, potentially some of the genetic information could get mixed and you could create brand new viruses that are a mixture of the two viruses, and potentially you could create a new strain of influenza A virus that has the capacity to infect humans. And pigs are believed to be one of the major animals in which brand new strains of influenza A virus that are capable of infecting humans can emerge. And that is why I have drawn this picture. We will come back to this later on when we discuss antigenic drift and antigenic shift. We'll do this in more detail, but know at this point that there is a huge number of different strains of influenza A viruses. Not actually that many infect humans compared to the number that infect birds. A huge number infect birds and don't infect humans. Most of the human strains have the serotype H1, H2, H3 as far as hemagglutinin is concerned, and N1 or N2 as far as neuraminidase is concerned. And the final message from this picture has to do with the pigs. There's a big overlap between strains that can infect humans and strains that can infect pigs. So lots of human strains can infect pigs, and there is a big overlap between the strains that can infect birds and the strains that can infect pigs. So a lot of bird viruses can infect pigs. So pigs could be infected by a human strain of influenza A virus and a bird strain of influenza A virus at the same time. This could create new influenza A viruses that potentially have the capacity to infect humans, and this could give rise to a whole new strain, therefore, of influenza A virus to which none of the population is immune, that could then cause a pandemic. And indeed, this is believed to be where new pandemic forms of influenza A virus emerge. So we'll have a break here. In the next video, we'll discuss the influenza B virus.